So welcome to the seminar number 17 of the International Tree Mortality Network. My name is Nadine Rühr and I'm very happy to see you all here today. I'm even more than happy to present to you Arthur Gessler today as our speaker. And I really have to say that Arthur is an amazing researcher. And I also think that for me, at least, he's really a true ecophysiologist. This is because he has integrated many different aspects into his research, from really very detailed plant physiological focusing on stable isotopes, for instance, during his time in Australia with Graham Farquhar. And now he's exploring more and more the ecosystem and forest dynamics aspect during his time at WSL. Since 2014, he is there at the Swiss Federal Institute for Forest, Snow and Landscape Research. He is the director of the long-term forest ecosystem research in Switzerland, and also the group head of forest growth and climate, as well as adjunct professor at ETH Zurich. So we are really happy to have Arthur here today. I'm sure he will give a wonderful talk. And also, I'm sure that you will have questions that I think it's really a good chance to ask these questions today. And you can do that either directly at the end of the talk, just unmute yourself and ask your question, or paste the question during this talk in the chat. And then we will read it out loud. Arthur, the floor is yours. Please.
Thanks, Nadine, for your introduction. Maybe you should not have raised expectations too high, because I won't talk about really everything, but because I have only 40 minutes or 35 minutes or so, but I will give you a kind of overview of new, as I think at least, new approaches for monitoring forest functionings in a changing climate. And let's cross scales from molecular mechanism to stand-wide processes. And certainly monitoring of functioning is broad. So I thought, we are in the Tree Mortality Network now. So the idea would be really, how can we get more information from forest monitoring on the risks of trees to die within a certain time period? So a little bit of how can we get forecasting information? And you all know that, but that's just an introduction you have to give when you give a talk in that network. Global mortality, forest mortality is certainly a global issue. And we have that in all regions of the world, also in obviously moist regions like moist tropics. And we have certainly knowledge that drought and increased temperature are the main drivers of that. And that certainly has also strong impacts on biosphere-atmosphere interaction. And here's only an example how from Bill's paper in 2016, how mortality changes carbon fluxes. So that's only that's not quantitatively, but that shows you just here you have a mortality event due to drought. And then certainly you have for a long time a reduction in net ecosystem productivity, which levels out depending on the intensity of the mortality event and depending certainly also on the forest ecosystems years later. And it's certainly not only the carbon sequestration which is impaired, which again feeds back to the atmospheric CO2, but it's on the other hand certainly also how water fluxes in the critical zones are affected. And it's definitely that after mortality event, runoff will increase with positive or negative consequences, just depending on the system you're looking at. And certainly the green water fluxes, transpiration will decrease. And again, it takes years to maybe even centuries if you have really stand replacing mortality until the situation levels out. So it's really important. We know that. And I don't tell you something new. But what was new for me, and Cornelius, who is also around here, he's a co-author on that paper, and Andy, who's also, I also have seen him among the audience. What was really new for me, that the disturbance is really strongly increasing. So I'm quite old in the meantime, and for me, 2003 was really the dry year and the driest year I have ever I have ever been exposed to in my life, and I thought, okay, that's now climate change. That's the extreme. And what you see here from Mauro Hermann in his paper, he looked at low NDVI anomalies in summer, so June, July, August, and they are displayed here in red in forest grid cells from 2002 to 2000 to 2020. Not 3, but 2 here. 2 is the latest. And you see here, and that is certainly something which is about the greenness, about the functioning, about photosynthetic capacity. And you see here, 2003, you don't see really anything, and also no legacy effects. But when you look at 2018, 19, 20, 22, you see a lot of red area, which tells you that disturbance is increasing more and more, which is certainly due to the hot and dry years, which are accumulating in the meantime. And climate science says this is a new normal situation. And I think that's where we definitely have to face much more disturbance in future. And now the idea was, and we are looking in my group and also in the program I'm leading, which is called Long-Term Forest Ecosystem Research, which is the Swiss part of ICP Forests. Do we find any early warning signals for tree mortality? We are mainly looking now at single tree mortality, but that is certainly also linked to stand level mortality. And the sketch I show you here, that's coming from medicine. Medicine is certainly also very much interested if you have some early warning signals, metabolites, genes that are expressed, which tell you if a person gets cancer in a few years, or if a transplanted organ is rejected, before you see anything really visible. And that's here the disease called disease phenotype. So you have a normal state, and then you have a transition state where you at the beginning don't see anything in the phenotype, and then you have a tipping point, and then you go to the disease state. And the idea is really to look for early warning biomarkers which tell you already at a stage where in the phenotype you don't see anything, that there is something happening. And that's the idea. Do we see, it's an analogy. Certainly you will see that we are certainly also looking at phenotypes, but do we see anything which can tell us that a tree, and that would be the transition state, dies? And so let's look for that. The first thing, certainly growth. Everybody who is working with trees looks at growth. And there is this very cool paper from Maxime Carrière and many co-authors published in 2017. And they compared growth signals in tree rings. And they compiled a whole lot of literature, and they looked at dying and surviving trees in the same stand. And then they try to extract information from that. And certainly then you have a dead or dying tree and you have a living tree. And then you can compare which information from the tree ring you can extract, and then look what is indicative or was indicative in the back look for mortality. And in that graph that kind of brings together a lot of information from that paper, you see here the growth ratio between dying and surviving trees. And that is certainly here, you see all the dying trees in the year before death, and all the dying trees certainly grew less in the year before death, but that's normal. Before you die, you normally have not the resources to even grow better than a healthy tree. But what is also important here, you see the duration of the period with either reduced or increased growth, and I will go more into detail in that later, before the tree died. So you can look back and see signals until 60 years before a mortality event happens. So there is something in the tree ring, which might tell you quite early that there is something wrong. And this gets together a little bit the information. So the scale is a bit different here. That's from a paper from us from 2018. So we say 20 years, but it could be certainly more. But you see, here is a kind of dying trees type 2. So they decrease. And remember, that's the either reduced or increased growth. The trees either start to decline in growth already quite early before they die. And that's the kind of baseline, the surviving trees here. So that is one type that you see. And the other type, you have often trees which grow even better before they die. And then they only decline very shortly before the mortality event. And we try to translate that into the carbon starvation hydraulic failure concept, which is certainly, it's a continuum and it's all linked to each other, and it's not black or white. But our suggestion was that when a tree really slowly decreases in growth and then dies slowly, that's more indicative of carbon starvation. That could be drought induced, that could be also competition related, different things. And if something like that happens, so a tree grows much better, that means it might need more resources because it's bigger and it's also taller if the diameter is larger, then it's more susceptible to hydraulic failure, as we know from a lot of different studies. And then this kind of hydraulic failure, or also pest and insect impacts, could then lead to that mortality trajectory. The problem is, I think there is a lot of information in the tree ring. The problem is it provides backward-looking information. So it's really a kind of a posteriori look. So you compare dying and surviving trees, and it's very difficult just to corner trees and to say, oh, there are indications that this tree will die, because you have to know which is a dying and which is the surviving tree. So for kind of looking back, that's provides you very important information. If you combine that with isotopes, for example, it also gives you physiological information you could derive from that, but it's not really a kind of early warning forecasting tool. But there are other approaches to look at growth, and that's from Roman Zweifel. He's a PI of the network called TreeNet in Switzerland. And you see here on that map, there are a lot of different points, and that are research plots where these point dendrometers are installed to trees. And you see here, it's a whole lot of different species. So it's over 50 sites in Switzerland, and the network is expanding now to other countries in Europe. And this system provides you really, because you get the information over wireless systems, you get really close to real-time measurements of growth, but also of the drought impact on the tree. And the drought impact is really important. So you see here, that's one of the point dendrometers, and you definitely see the resolution for the measurement is so high that you really see changes in tree circumference or diameter, as you want to express it, in the micrometer range. That means you see the daily shrinking during daytime and then swelling of the tree, which is related to the water capacitor of the stem, which is water storage in the bark and in the wood, which is kind of used for transpiration during the day when transpiration is higher than root uptake, and when this capacitor is refilled via root uptake in the night when transpiration is low. So that's what you see here, that are these daily variations. And oops, I have to go back, sorry. There was something strange happening. And you see here, there are periods when the diameter of a tree is not expanding. So that's really what, in principle, the system is measuring. The tree is even shrinking over several days. And that's a period where the water that is spent by the tree via transpiration, the amount of water is lower than the amount of water that can be taken up from the soil. That is happening when you have really high water pressure deficit in the air, but only low water supply. And during that time period, we define also the tree does not grow. And only if you have then a rainfall event, then you see that the stem expands irreversible, and that's a growth period. So we can, and this, the area between this line and this curve, that is what we call tree water deficit. And that's a measurement for the stress that trees are exposed to during drought. And we are just about to try to translate that information also in water potential, but that is not what is really achieved at the moment. But that's certainly then a drought indicator, and it's a growth indicator. And how does our system now display that? So we have, on the one hand, signals in growth, and we have signals in tree water deficit. And you see here the map, that's a tree water deficit. And you see here, these points show the different plots and sides of TreeNet. And you see here, the water stress of the tree is always high when you have here very pinkish dots. And what is in the background is modeled soil moisture deficit. So that's a hydrological parameter we model at WSL for whole Switzerland. And then you can really see how the soil hydrological situation looks like and how the trees react upon. And the same you can certainly, because I said you can extract on the one hand this tree water deficit, and on the other hand, over the longer time period, the irreversible expansion of the stem, which is defined as growth. And you can look at that, and that's just from a given time point last year in August 2022. Okay, and that allows you certainly then to look also to compare the growth trajectory of the given situation, the given year. So that's really the months, and that's the year 2022, and that is the growth in that year of the tree. And here, the black line and the shaded area, you can see the average growth over long time periods. That means in that case, it might be 7 years or something like that. So you can really now compare how growth looks like. And you can certainly also, because we said it's close to real-time information, you can then certainly also look how the, on the one hand, water deficit in the background modeled, but then also in the foreground, how the tree react to that water availability. And that then allows you certainly to get long-term information on the short-term impacts of the current climatic or water availability situation on tree water stress. And that helps us then to define areas at risk where we have to expect mortality. And we defined such areas at risk, partially certainly from the tree network, but also from other observations, which I will go into detail closer later. So, and you see here the Valais, it's the Rhine, the Rhone Valley in Switzerland, which is really a very dry inner alpine valley. And we have a lot of different monitoring sites here. And we have also the TreeNet sites here. And here, it's definitely from the TreeNet data that trees are suffering from water stress continuously in summer. And on these sites, this Finwald, Lens, and also Saillon, we also observe high mortality rates of these trees. And you see here, that's the summer precipitation, which is shown here. And the hotspot of mortality is here in the area around Wisp, and we have very, very low summer rainfall here, and the mortality is lower in that direction. So that kind of system might now not only tell us about the current status of the water supply and of the water stress of the tree, but if we look at that continuously over the years, we also see areas where trees are regularly prone, and then we can compare that to mortality. So that might be one tool to understand where forests are at risk and where we have to look closer what happens and if the trees are in future potentially or already now subjected to mortality. So nowcasting provides real-time data on the response of trees to heat and drought, and it allows to identify areas at risk. We can't now say a tree that has a particular tree water deficit will die soon, but at least we can define this area where we have regularly strong drought effects. That is an area where we have a risk of mortality. I want to go now to something which has been done since the 1980s of the last century within the framework of the forest decline, which was really Waldsterben was certainly something when I was at school, that was a very hot topic and everybody knew about that. And many of you might have been young at the time too, and they can feel what that meant. We were all, or many people were thinking, okay, there will be no forests in future anymore. And at that time, it was clear we need a monitoring system, a Europe-wide monitoring system to understand the health and the vitality of trees all over Europe. And at that time, ICP Forest was set into place, and that guaranteed a kind of continuous and also homogeneous assessment of tree vitality via assessing defoliation all over Europe. And you see here examples of spruce trees. Here it's a, let's say 5% defoliated. It might not be really an ideal 100% foliated tree. And here you see quite some defoliation. And that are field crews which are going into the field, looking with binoculars in the crown and assessing the defoliation of trees. And you see here from 2021, a map of defoliation. That's the mean plot defoliation of all species in 2021. So that might give you now a first impression, okay, defoliation, that is something that might be related to the health status of the tree, and that might be related to maybe future mortality. So we have to look a little bit closer into that. So that's again on one of our intensive monitoring sites in the Valais, where I told you it's an area at risk, and we have already seen mortality there, and we assume that we have more mortality in future. And you have here in the bold black line, that's the annual mortality. And you see we have mortality peaks here, we have mortality peaks here, we have mortality peaks here after 2003, for example. And it's often that you see these mortality peaks a year later. You have 2003 was hot, dry year, and the mortality peak occurs in 2004. And what you also see is that you have a comparable signal in the annual defoliation change. So that looks like as if defoliation and the change in defoliation in a forest is related to mortality. So that now looks like you have a one-to-one situation. So you have high defoliation and at the same time you have high mortality. But it looks, in principle, when you have a closer look, it's a bit more complicated. So we looked at, in total, 900 trees, and we binned them in different defoliation classes. So that is now very healthy tree from 0 to 22% defoliation. And that is really a very, not healthy tree with very high defoliation. And then we look at these trees and how their future trajectory was. And what you see here, that's 1, 2, 3 to 6 years. And that's the mortality rate within 1, 2, 3 or 4, 5, 6 years. And what you see is a very highly defoliated tree, has a probability of more than 80% to die within 6 years. And even a tree with it's a relatively strong defoliation anyway, has a risk to die within the next 6 years of almost 40%. So that means defoliation of single trees allows to estimate mortality probabilities. And that is for Scots pine. And we are now looking at many other tree species within the ICP Forest network. And there are comparable signals, but the kind of time to mortality might vary from species to species. So that means here, with the defoliation assessment, which is something that is done on thousands of plots all over Europe regularly every year, that the assessment of defoliation trajectories allows to quantify the mortality risk in the next 1 to 6 years. So maybe that's a short period, but that is really something which allows us, on the one hand, to potentially link to remote sensing, because it's a parameter which is related to the canopy, and you might be able to see that from above. And that at least gives you some time period for a kind of early warning risk detection. We thought, okay, let's go a little bit more into the mechanisms. It's the same field site, it's Scots pine, it's the same defoliation classes. So we have here from 0 to 20, that's the healthy tree, that's the baseline. And then we compared increasing defoliation, again, binned into classes. We compared the metabolites in the leaves of these different defoliation classes to the very healthy control with almost no defoliation. And that sounds now like a very complicated graph. And don't be afraid, I won't go in any details and I won't plot you any metabolic pathways. You just have to look at the heat map. So it looks like that here, compared to the baseline, the healthy tree, not really much is happening. You don't have a lot of colors. So mostly everything is white, and here the same. And only when you go now to higher defoliation, you see changes in the patterns in the heat map. And it looks like there is a kind of tipping point. A tipping point when a tree has lost more than 80% of its needles. In the case of Scots pine, the metabolic profile looks different. Okay, that is now a very general pattern without going into details. So tipping point changes in the metabolites in the leaves. And that is a classical medical early warning signals. In medicine, people like to have metabolites or something like that as markers for seeing changes in metabolism and therefore kind of disease warning signals. We looked a little bit closer. We thought now, what happens and which metabolites are really changing? And it's a like of zooming in here, the needles. That's again this class here. That's the class where we see a lot of differences compared to the very healthy trees. And just to mention, that's a log ratio changes. So it's the relative changes, but then put on a logarithmic scale just to compress the scale a little bit, so that really strong changes are not kind of masking only slight changes. So that's just the only reason for that. It's just a comparison with the healthy trees. And that's the same way how we compare it now. It's also log ratio changes. And that is the high defoliation class, again, compared to the healthy trees. And you see here the needles, which I showed also in that heat map. And you see here the fine roots of the same trees. And that are really the same, the fine roots belonging to the tree, because we had a tractor which was pulling down the trees so that we really could access exactly the fine roots of these trees. And not just digging in the soil and maybe the roots are coming from a totally different tree. And I think the interesting thing that what you see here is in the needles, a lot of stress-related metabolites are increased in their concentration. So osmoprotectants are mostly increased. Defense-related precursors are mostly increased, with one exception, which is, we don't know why, coumaric acid. Antioxidants are either not changed or they are increased. So that looks like the tree is still trying to keep the functioning of the needles continue to work. But when you look at the fine roots of these damaged trees, then you see all protecting, defending compounds, they are downregulated. It looks like that the tree is really starving from metabolites, from carbon compounds in the roots. And it looks like that these trees, which are, and I told you these 80% trees are most likely to die within the next 6 years, they die from the roots because the roots are not supported any longer with the metabolites which are needed to defend themselves against pathogens, to provide really avoidance of antioxidative damage. And the tree is really dying, obviously, from its roots when you look at the metabolite fingerprint. So that means metabolic markers, they provide mechanistic information, and they are related to the mortality risk by defoliation, because I want to go back here. You see the very highly defoliated tree class is most likely to die. And there is a very big step to the next defoliation class. And it's really the same here. You have a very strong metabolic signal in exactly this group, which is very likely to die within the next few years. So that's really fitting together. Okay, and then we thought, yeah, is there another possibility related to leaf trait information like defoliation, but also like the metabolites, which can be used from the remote sensing side? And that was now work of Petra Dodorico, which published that paper in Plant Cell Environment a couple of years ago. And we focus now on the xanthophyll cycle. We know the xanthophyll cycle is really very important to quench excess light energy, which can, under particular stressful circumstances, not used by the Kelvin cycle. When you look, for example, at a tree under drought stress, the tree closes its stomates. The light energy is still there. And then the closing of the stomates causes that no CO2 can be taken up. So the Kelvin cycle has no substrate. And the light energy is still there. And this energy is potentially dangerous because it can destroy membranes, it can oxidize proteins. So it has to be quenched. And that works via the de-epoxidation of the xanthophyll cycle. So the level, on the one hand, the level of carotenoids, where the xanthophyll belong to, is indicative for these stress defense mechanism, and also the status, if you have now more violaxanthine or more zeaxanthine, that is really important. And if you have a high stress level, you have a higher de-epoxidation of your xanthophyll cycle. And that is kind of mirrored in the photochemical reflectance index, which definition is here. So you need a multispectral sensor, and then you target for these particular two wavelengths, and this PRI gives you information on the status of the xanthophyll cycle. That means how much stress or how much stress the plants are exposed to relating to excess light energy. It's a general stress parameter, but under drought, it's certainly mostly related to the non-availability of substrate for photosynthesis. Okay, so let's have a look. What can you do with that? And I have to go a little bit. I have a little bit of time left, and I promise you I come to the end quite soon. So that's the view from above. So the left-hand picture of our experimental site, Fienwald. Fienwald is a pine forest, Scots pine forest stand, which is very dry, which is close to the dry edge of distribution of Scots pine. And here, the red squares around the trees, and that are single trees, single canopies of single trees, that's our control site. So we assume it's very dry, and trees are in summer quite drought exposed. And you have here these blue squares. They are smaller than the red ones. That has a reason. And that is our irrigation treatment. So we add to that very dry trees double of the amount of the annual precipitation. So annual precipitation is over the long time period 600 millimeters. And so these trees, they get 1,200 millimeters. And these yellowish bars, that is our irrigation stop site. So irrigation has started 2003 and is continued. Irrigation stop, irrigation also started in 2003 and has been stopped in 2013, 10 years later. And we are now looking at the legacy effects. And that is replicated in a random block. Now, these are not random because the irrigation stop had certainly to be performed on the irrigation treatments, but the control and the irrigation blocks, they are in a random design. And that is now an image that's also from a drone. That's from a DJI drone. We flew several times over the year over the Fienwald, and that's now the PRI image. That already shows you, okay, here more reddish, here less reddish. And that is now aggregated on the crown level. And you definitely can easily see which are the control sites, the red ones. And they have a very low photochemical reflectance index that tells us that, or we assume that is a proxy for a strong deepoxidation of the xanthophyll cycle. And here you see the bluish color, where we have a much higher PRI, and that's the irrigated sites. And somewhere in between is our irrigation stop site. They received at that time already 8 years no water, and they are still intermediate. I think that's really interesting, and we have until now not a very clear explanation for that. But what we certainly did, we also measured ground truthing parameters like pigments, and that's a deepoxidation status of the xanthophylls. And there is certainly a clear significant relationship. And we also looked at chlorophyll fluorescence, because that's certainly also, you can derive a parameter that's called non-photochemical quenching, and that's really a measure for the portion of the light energy which is quenched away as heat. And you see for different years, it's a little bit different, but you see also a clear relationship in between. So that means the reflectance index tells you something about the stress level of the trees. And at the moment, we are really about to link now at different sites longer-term observations of this PRI. That means a pre-visual stress level, because you see that only when you look at these particular wavelengths, it's not really visible as bleaching or as other things. These pre-visual signals, we try now to link to trajectories, longer-term trajectories of defoliation. And our hope is that we can, with all these different parameters together, get a kind of framework of early warning signals for tree mortality. So we have understood now that our nowcasting network, which we want to expand over larger parts of Europe, together with classical forest monitoring, together with satellite remote sensing, allows us to define areas at risk. And then really also allows us a kind of nowcasting of the current stress level trees are exposed to in a region. Then the idea is with the PRI, and if we could then link that, and that's still work in progress. With a PRI, we get a kind of very early pre-visual stress signal. And if that is reoccurring ever and ever, then this might translate into defoliation. And we see that defoliation and the metabolome seem to be linked. And the metabolome is indicating that when we look at the roots, at least, that the roots are not supported sufficiently with the metabolites they need for defense and for other important basic metabolic mechanisms. And then we already have seen that from defoliation, we can predict the mortality risk in future. And I think the earlier we understand which parameters tell us in the end mortality, the better we can predict which areas are at risk. And that's certainly different from agriculture, where you can quickly change your management or the crop choice or your rotation scheme. But still management interventions might be possible, like thinning, already starting to promote other more resistant species. And we are now talking about time periods of maybe 10 to 15 years, which is really not long for the rotation time of a forest. But that means we need to strive for even getting more information, which might tell us even earlier what is the long-term trajectory of a stand. But to summarize, there are potential early warning signals for tree mortality. On the one hand, the defoliation trajectories, and the nice thing is that is measured anyway at many, many sites in Europe. They help to explain the mortality risk in a time frame of, let's say, 10 years. The metabolite homeostasis, that allows us to change, maybe together with defoliation, mortality causes, and with regularly pre-visual stress assessments, like reflectance indices, we might even get earlier information, and if linked to defoliation, that might allow us really to look maybe not only 10, but 20 years ahead. And that is really the main message I want to tell you. We are looking for that, and I think that needs certainly then, as I said, combined with classical monitoring, with remote sensing, and then we might better understand what the fate of given forest stands, of given trees in given regions of the world are. Thanks a lot for your attention.
Thank you very much, Arthur. It's really impressive seeing that you're in Switzerland already are able to monitor forest health in real time. I'm sure there are plenty of questions. If you want to ask Arthur a question, please raise your hand or use the chat. I would start. I think you already answered one of my questions on the forehand. You showed before that we see that growth decline for some of the trees 20 years ago, up to 60 years ago. Mm-hmm. Do I get you right that you would recommend to harvest these trees directly, also for competition release? They will die anyway, so why not taking them out already now?
Yeah, maybe that's a good way, but the question is now, if you always compare here with the surviving trees, but when you now measure trees, do you know if this one will survive or that one will survive? Or because that is certainly, that's a baseline, and that's a very virtual baseline. You have a whole range of growth rates, and you can, once you know that a tree died, it's easy to say, okay, I can look back and I see this growth pattern. But if you now look at the growth pattern of a tree here at that time point, here you might take out these ones and not this one. So the question is really that looking back is always very clear. You know which tree is dead. You know that you can also derive then reasons for why a tree maybe died. But if you really only look at growth pattern for some time, it's very difficult to predict if that tree will survive or not. I think that's the main question.
Yeah. So still no hands up. I was also very impressed that you showed, you're kind of building the bridge between the carbon and hydraulic community, that you showed that in roots, the metabolites are downregulated. You mentioned that it was related to the carbon balance. Do you have any data on that?
Yeah, we measured certainly also the whole tree 13C allocation in other trees, not in these ones. But we know that when trees get drought exposed, they are not able to allocate carbon to the roots anymore. And we see that especially because it's a Pien-Wald experiment here. And the design is that we have irrigated and non-irrigated dry control plots. And when we compare, then we definitely see that the allocation in the dry control plots to the roots, the carbon allocation of new assimilates is much lower. And that over time translates also in the kind of root projection area to be smaller. That means it has not only this short-term effect, there is less carbon allocated, that causes also a long-term effect that the rooting system gets smaller. So there seems to be, and when we look now at these dying trees or at these very strongly defoliated trees, it looks like that this has then also an effect for the root metabolites. It's very difficult to look at it's a hen and egg system, because when it's dry, then certainly the conditions for root rhizosphere metabolism are not good, and then the zinc strength decreases, and then that might be a reason. But on the other hand, it might also be a reason that transport is impaired, and that really not over the longer term, not enough carbon is then reaching the roots.
Thank you. We have a question by Mirella Belui. Sorry if I didn't pronounce it correctly. Thank you very much for the talk. Could we say that we are already able to predict tree mortality if tree level defoliation is quantified by remote sensing? Or would we need more information?
Yeah, I think that's what we at the moment are looking at. Because the important thing is we do have this ICP forest information that is really looked with binoculars. You look from the ground to the canopy, and at the moment we are using drone-based assessments at the same sites in Switzerland where we are doing the annual defoliation measurements. We fly with drones over the same sites and we assess from above the defoliation. And we now first have to look if that fits together, because the view is totally different. And it looks like, at least for some tree species, it can be easily compared. For others, it might be more difficult. But if we have that, then we can certainly translate into airplane-based remote sensing, for example, and then get information on the area scale defoliation, and that might then be used to assess mortality risks. But I think there are still some steps we have to get over until we definitely can say remote sensing allows us to predict mortality risks.
So Asta is raising her hand, please.
Yeah, thank you. Can you hear me?
Yes.
Yes.
Yeah, thank you, Arthur. Sorry, I cannot open my video. I'm not in a good place, so I will just ask. Thank you, Arthur. It's really inspiring talk as usual. My question is that you said tree rings is only for, after the tree is dead or it's dying. But if you take samples, for example, in 2003, we took samples and you can see the recovery, the resilience of that species, especially when you compare different species, right?
Yeah.
And when you combine it with the isotopes, like the carbon and oxygen, can tell you about where water is coming from, and its drought tolerance. And you can compare and characterize species. And you can say, okay, if the environment is going to get dry or warm, maybe it's better not to plant this species in the future. So in a way, aren't we avoiding or minimizing, the happening of tree mortality? Isn't it by itself early warning signal.
Yeah, I think you're right. Certainly, if you definitely see that a species is not able to recover very well from a drought event, then certainly it's already telling us that this species might not be able to grow there in future. And maybe I was too negative about the tree rings, because I think what is really important is tree rings can tell us certainly also something about, as you said, the kind of resilience, how fast trees can go back in growth, or also in physiology, when we look at the isotopes, to their pre-stress state. And if you have sufficient information, you might be able to understand how different recovery trajectories translate into kind of state changes. And we are looking at these state changes, and we're also looking at tree rings. And if recovery gets slower and slower for a given species, for example, under different conditions, that might tell you that this species is approaching a kind of tipping point where you have it's often called range or state shift. And for a tree, that means certainly, for an ecosystem, that means a forest is changing into a steppe or grassland. And for a tree, it certainly means it's transiting from a living to a dying state. And I think this information we can extract, and I think we have to learn a little bit more about how temporal trajectories of recovery can be indicative for also assessing mortality risk. I think there are different ways to approach that, certainly.
Yeah, thank you.
Thank you, Arthur. Tom, please.
Hi, Arthur. Really nice talk. I'm just kind of reflecting on the results of Maxime and the plot you have on the screen, with these really long lags for some trees between really long reductions in growth rates before dying under drought. And have you looked at all at kind of what fraction of this mortality then might be mortality brought forward by the drought, something that was going to die anyway in the next few years, but it's just kind of being concentrated into a small period versus? Yeah,
exactly. I think I don't think Maxime looked into that, and we also in that paper did not look into that. So it's certainly something you will have during drought periods excess mortality, which is higher than kind of baseline mortality due to self-thinning. And then in the year thereafter, you might have even lower mortalities. And I think that is something it needs definitely to be looked at. Because we are not talking about stand replacing mortality mostly in these cases, because there is always a comparison between still living trees and dying trees. So it could really be that a drought event just wipes out the trees which would have been dying anyway. And especially these slow trajectories, they might tell us about suppression over longer time. And then it's just that the resources in a given year are not sufficient to maintain the carrying capacity of a system, and then the tree dies. And this not having enough resources would have happened anyway years later. I think it's really important to look into that. I fully agree.
And do you have an idea how you might do it? I mean, can you do this from a remote sensing, or is it always the trees under the canopy that are the ones that you're seeing those signals in?
Yeah, I think that's a little bit tricky. I think you should maybe, one should really look at long-term forest trajectories. Therefore, you need long-term monitoring data. Define the self-thinning line for these over the longer term, and then look if the trees which you are now looking at, if they were kind of causing excess mortality over the self-thinning line, and even the years later, then there is lower mortality than expected from self-thinning. I think that would be the optimal way to go for, but therefore you need very long-term observations for many sites, and you don't need interventions, which is very difficult because in the managed forests we do have in Central Europe, it's almost impossible to assess that. So can't say how to best approach that.
Thanks very much.
Another growth-related question. You showed that in 2022, according to the TreeNet data, growth was kind of almost no growth, right? Did you observe strong species differences?
Yeah, we observed species differences, and oak trees were growing better. But I think Roman would be the one to answer that, but I think he is not around here in the audience. Oak trees grew better, and it was certainly very much also dependent on the stand, where the stand was. We have increase in growth level at high altitude, and even for the same species. When you look at, for example, spruce suffered strongly in the lower areas, and it benefited even from the hot and dry conditions in high altitudes. And there certainly we have to assume that it's the temperature limitation that you have in high altitudes. The problem with spruce, however, is really the bark beetle now, and the generations of bark beetles, they get now 3 to 4 generations, even at 1,500 meters, which still threatens spruce, even so they are really in their normal distribution range. So I'm not sure what will happen in future with spruce in the higher altitudes. But at that time, growth was increased.
Cornelius, please.
Yes, thanks, Arthur, for the nice talk. Well, as a remote sensing person, I have to ask a remote sensing question, of course. I was wondering, the index you develop, you really need like kind of narrow band hyperspectral data for this, and we do not really have this from space. So have you compared it also to more common indices we can derive from spaceborne remote sensing?
I think PRI is a very common index. No, but I know it's not in the Sentinel portfolio. I think, no, we did not. But I think we will do that because we are really looking now at different indices and how they are related to our defoliation. The PRI, me as a plant physiologist, for me, PRI is certainly what comes closest to chlorophyll fluorescence and gas exchange measurements. And we also looked at the chlorophyll carotenoid index, which is not as good explaining this stress levels we saw at Pinwald. But I think definitely other indices will be compared with the defoliation, first on a drone level. So we are looking now at visual images, and we are looking at NDVI, and we are certainly still looking at PRI. We are also looking at thermal imagery, because that might also provide us information on transpiration, which is then certainly also related to either heat stress and also to the potential of stomatal closure and therefore changes in PRI. But certainly we have to look at other indices which are more common for satellite remote sensing.
So thank you very much. I think if there are no further questions, I would close the session. Once more, Arthur, thanks a lot for this. It was a pleasure. And also, of course, for the audience, and see you next time.
Bye-bye.